Well, good evening and welcome to the 2021 Lake Huron Regional Fisheries Workshops. We're excited and appreciate everybody taking their time to be with us this evening to chat for a couple hours about our amazing Lake Huron fisheries, and on a perfect day, Earth Day, to have that conversation. So glad each of you have taken some time out of your busy schedules to hang out and talk fish this evening. So we have a super exciting agenda this evening. I just wanted to hang on this introduction agenda outline so you can see some of the talks that we can look forward to. We're going to hear about our Lake Huron prey fish, an exciting Lake Huron, Lake Michigan predator diet study. Here's some offshore fishery updates from the Michigan DNR. We're going to learn from Pete Esselman with the USGS Great Lakes Science Center about a really neat and innovative round goby monitoring effort, and then we'll round out the evening with some opportunity to hear and visit about some of the Lake Huron management updates. I just wanted to say this is a traditional each spring meeting. It's kind of a health check, if you will, on our Lake Huron fishery. It's a great opportunity to bring researchers and managers together with anglers, communities, or anyone involved or simply interested in our Great Lakes Lake Huron fishery. So we do this each spring. Typically, we do them out in the communities, but given the times, we're having a virtual workshop again here in 2021. So we're kicking off this series with the offshore open water fisheries focus this evening. But I wanted to share that next Thursday, 3 Thursdays of Talking Lake Huron, next Thursday we'll shift to a nearshore focus where we'll talk more about Saginaw Bay, the Relational Islands area, the St. Mary's River, touch on some Cisco restoration effort updates and cormorant management updates, among others. So if you're excited about the nearshore fisheries, join us next week. And if you haven't had enough of Lake Huron fisheries, you can join the DNR Conversation and Coffee on May 6th to have a chance to ask questions and learn more from the DNR fisheries. And Michigan Sea Grant is excited to be a partner in that effort too. So if you don't get your questions answered tonight, or you have things that you want to bring up at a later time, that would be a great opportunity to have that personal conversation with the Department of Natural Resources. Before I move on, I like to start these workshops with thank yous. There's a lot of partners and contributors that pull together to make these workshops possible. And I think it's important to acknowledge that this is truly a collective effort to bring these education programs together. So from an agency standpoint, a research and management standpoint, we have our Michigan Department of Natural Resources Fisheries partners, our USGS Great Lakes Science Center partners, U.S. Fish and Wildlife Service partners, the Eagle Office of the Great Lakes, and our very own Michigan State University Department of Fisheries and Wildlife, among others. And many community partners and organizations and members from those organizations have contributed to these workshops over the years and help to get the word out to make sure that folks have the opportunity to participate in these sessions. So those are partners like the Michigan Charter Boat Association, the Michigan Steelhead and Salmon Fishermen's Association, the Blue Water Sport Fishing Association, Hammond Bay Area Anglers, Lake Huron Sport Fishing, Saginaw Bay Walleye Club, the Thunder Bay Walleye Club, Thunder Bay Steelheaders, and the Detroit area steelheaders, among others. A lot of great community partners that have helped over the years and this year to bring these workshops out into our communities. And I need to end with a mention of the Lake Huron, the DNR Lake Huron Citizen Fishery Advisory Committee. It's worth noting that this committee, and many on this call are members of that committee, gathers quarterly and contributes important input and ideas as advisors to the Department of Natural Resources Fisheries. But I also view those advisors as Michigan Sea Grant advisors. They spend a lot of time helping us to think about what we want to bring out to the communities through these educational workshops. Frank Christ is on the call. Frank is the chair of that committee. Frank and I will be listening and learning along with you all and be sharing updates and a report back to that advisory committee out of this conversation. And then I need to end on, my own awesome Michigan Sea Grant team. So I am Brandon Schroeder. I'm an extension educator for Northeast Michigan, Northern Lake Huron, serving out of Alpena County. But I wanted to introduce some of my other Lake Huron focused colleagues, Elliot Nelson in the Eastern Upper Peninsula, Megan Goss, who serves the Saginaw Bay region, and then Mary Bowling, who covers our Southeast Michigan coastlines and splits her time between Lake Erie and the far south of Lake Huron. And then our fantastic communication team, Cindy Hudson is on the call in the background, helping to make sure that we are moving along and everybody's able to stay connected as we have this virtual session. Just a quick plug and pitch for our Michigan Sea Grant program. Michigan Sea Grant is a program of NOAA. And so we're a coastal ocean science focused program. And in the Great Lakes, our oceans are really this unique freshwater seas we call the Great Lakes. So no salt, but still invaluable resources to our state and our country and our world, with 20% of the world's fresh water available to us on the surface of the planet. And our role is really to think about how to support Great Lakes science and research and make that research useful in our coastal communities, and so that we are able to sustainably use these resources for the social, ecological, and economic benefits they provide us. So many areas of work that our Michigan Sea Grant program dabbles in. We spend energy emphasis working in resilient coastal communities and economies. We invest in healthy coastal ecosystem work, and we invest in a lot of environmental literacy, Great Lakes literacy, and education efforts. But tonight we're here to talk fish, and so a big part of our work is really looking at the sustainable fisheries that we have to benefit from in Michigan. So I mentioned Michigan Sea Grant promoting Great Lakes science and research, and then trying to make that useful in our coastal communities and folks that participate in the fishery. So I just wanted to make a note that there's some great resources we offer. Make sure to check out our website. We've got some nice fisheries focused publications like The Life of the Lakes, a guide to the Great Lakes fishery, and then a salmonid identification guide that came out last year, which is a nice reference and resource that you'll probably want to have on file. And I think in sort of closing up my introductions here, I just want to point out this anti-discrimination acknowledgement, because we receive federal funds. We have we were required to, but we also have made a commitment to ensure that our programs and resources are accessible to any and all who would value or benefit from those resources without discrimination. So we've made that commitment, and I hope you all will help to share these resources widely among any and all who will value and benefit from the work and resources and programs we provide. And I will get us onto our agenda. I'm going to introduce Tim O'Brien, who's with USGS Great Lakes Science Center. They've been a longtime partner in these educational efforts, and they really are at the heart of the monitoring of the prey fish species in Lake Huron. So it's really nice each year to get that status and check in on what's going on with the Lake Huron prey fish community. So with that, Tim, welcome.
Hello and good evening. My name is Timothy O'Brien. I'm a fisheries biologist at the USGS Great Lakes Science Center. Tonight, I'd like to speak with you about offshore prey fish populations in Lake Huron. Just first, some background. Prey fish are important to the Great Lakes for a number of different reasons. First off, they provide food for economically valuable fisheries, both sport and commercial. They represent opportunities for commercial fisheries themselves. So we think about Cisco and lake whitefish. When these fish are juveniles, they serve as forage. And then also species like rainbow smelt have been harvested commercially in the Great Lakes. Third, we also think of prey fish as an important part of the Great Lakes in terms of the health of the Great Lakes ecosystem. And in particular, we think about energy transfer from lower to higher trophic levels. A recent project funded by the Great Lakes Fishery Commission provides some materials that illustrate this nicely. Just seeing here this miniature food web model, and we can see prey fish sort of occupying this middle position of the flow of energy. So as nutrients come from uh, either benthic or other organisms, algae, and these nutrients are then brought up through the food chain, prey fish serve sort of that important role to shuttle them further up towards top predators. And then just showing here on this corresponding energy pyramid, we see alewife and bloater and other small prey fish occupying trophic levels 3 and 4. USGS prey fish assessments use a couple of different tools to describe the spatial and temporal trends in relative abundance and species composition of prey fish throughout the lake. We have two different surveys, the bottom trawl and the acoustic and midwater trawl surveys. These surveys target different fish guilds, so both demersal or bottom-oriented, as well as pelagic or semi-pelagic fishes that are suspended in the water column. They also cover different time periods and different portions of the lake. Acoustics is considered our whole lake survey. This began in 2004, and this covers all of the basins of Lake Huron, the exception of 2020, as we'll see here in a minute. The bottom trawl survey is the main basin only, but it's a much longer time series, now exceeding 4 decades. So we have these two different tools, and we look at the results from them as complementary. And then just to illustrate the two different methods that we use for surveying prey fish, shown here on the left is the midwater trawl. So this is then, as the name implies, towed through the middle of the water column or sort of somewhere here other than the bottom, wherever fish are distributed and suspended. We use this in combination with the acoustics and our sonar data here that we're collecting. It's just showing a representation of the sonar beam. So these two are deployed simultaneously. And then contrasted this with bottom trawling, which occurs during the daytime. The trawl here being dragged along the bottom of the lake where fish are aggregated during daytime. And acoustics then occurring at night. This illustration on the right also shows some of the reasons that two trawl surveys are needed. Often fish are distributed above the trawl path. So these are fish that are in the bottom trawl blind zone. They're not available to the trawl. And acoustics also cannot pick up all the fish. So really these two methods, in theory, kind of balance each other out. In 2020, it's important to point out that USGS assessments looked quite a bit different. We did have no sampling in the Canadian waters of Lake Huron in 2020, and the acoustic survey greatly reduced in spatial scope and length as a result. And then the bottom trawl survey was nearly completed, except again in Canada, our single port in southern Lake Huron off the port of Goderich was not sampled. But this led then to a situation where, for the first time in several years, both the bottom trawl and the acoustic survey essentially covered the same portion of the lake. And that's shown here in this map. Our standard bottom trawl locations are shown here at the ports of Harbor Beach. These are the open circles. Au Sable Point, Alpena, Thunder Bay, Hammond Bay, and Detour. And then the acoustic transects and midwater trawls shown here, black line and triangle. So you can give a pretty good idea of what kind of coverage we had in the lake in 2020. So just to start out with the results. So in this presentation, we're going to report on long-term shifts in species composition. And for this, we're going to use the bottom trawl survey, spatial trends in abundance, and for this we'll use the acoustic survey, which is again traditionally whole lake. This type of data lends itself well to looking at spatial trends. And then trends in the abundance and composition of dominant species since 2004. So bloater, rainbow smelt, and alewife are primary species. We use both surveys. Cisco only from the acoustic survey. And then sculpin and round goby are bottom fishes from the bottom trawl only. And then just looking at the trends for some of the more common species that we see in the offshore. These slides are going to be oriented pretty similarly. We have biomass of large fish at the top, density of young fish. These are the newly produced recruits for a given year on the bottom. And really, if we look at bloater, the last couple years, decade to decade and a half, we've tended to see this increase in biomass. We've also seen an increase in small fish production. When we look at the two surveys overlaid, so again, this is just 2004 through 2020, looking at bottom trawl and acoustics, they track pretty well. We've seen a couple years where the bottom trawl diverged quite a bit, but generally we've seen this increasing number in biomass of large fish, and pretty variable, but generally it's kind of an increasing trend in the small fish as well. In 2020, most of the bloater were found in the northern portion of the main basin, shown here. Size distribution between both the bottom trawl and the midwater trawl survey was pretty similar. Some smaller fish picked up in the bottom trawl survey, but generally a similar size range. These are pretty small, even for adult bloater, in how we think of sort of a marketable sized bloater that the commercial fishery might harvest. Rainbow smelt, also an important fish in the offshore waters of Lake Huron, really abundant during the 1980s and early 90s, and then we started to see this sharp decline occurring. In recent years, we have seen more increased production of young smelt, but generally much lower biomass. If you look at the two surveys overlapped, a little bit noisier here. We see the bottom trawl tends to, in a lot of cases, pick up more fish in terms of rainbow smelt, but generally they track fairly well in terms of the general trends. Young a year fish also fairly well. Primarily in recent years, we see pretty similar trends between the two surveys. In 2020, rainbow smelt were primarily distributed in the central and southern portions of the Michigan waters of the main basin. The lengths distribution between the two surveys was very similar. We tended to see sort of a nice split here, where we saw young of year fish, and then sort of the older component of the population. Pretty similar size measured by both surveys. Looking at alewife, this is a really familiar story for most everyone. Certainly high numbers of alewife during the 1980s, 1990s, and then this collapse that we saw early 2000s occurring right around the time that we started to see some major food web changes in Lake Huron as well, related to nutrients and lower food web. But this story has remained the same really for the last 15 years or so. We've seen very low levels of alewife. We have seen occasionally some production of young fish, but certainly much lower than previous years. And similar to rainbow smelt, this has not turned into any notable increase in adult biomass. We see really similar patterns when we look at the two surveys overlapped for the main basin. The bottom trawl does tend to pick up a little bit more in terms of the larger alewife, certainly, and also in the case of the young of year alewife. That being said, we can see by the scale here that these numbers are still extremely low. There is still some small levels of production that we see occasionally for alewife in Lake Huron. In 2020, most of the alewife were distributed here outside of Thunder Bay. In the last couple of years, we've tended to see this. Alewife have been distributed sort of in this portion of Lake Huron. Uh, very low numbers in 2020, so we really only saw this little pocket. The length of these fish ranged anywhere from 30 millimeters. We did see a few larger individuals in the bottom trawl, but the majority of these fish, incredibly small size, young of year fish. So all of these were aged at age 0. Adding in a little information here on Cisco because of their importance to the ecosystem and recent efforts to stock Cisco as a prey fish, and then also develop a fishery for Cisco in Lake Huron, particularly in Saginaw Bay. If we look at current distribution of Cisco in Lake Huron, and this is based on acoustic midwater trawl estimates, Georgian Bay, North Channel, and the northern rim here of the main basin is where we see current populations of Cisco. And I should point out that these are primarily, these are adult Cisco. It's the corresponding lengths from roughly 200 millimeters all the way up to close to 500 millimeters. So these are larger fish that are caught during late summer, early fall acoustic surveys. And they're foraging off in the epilimnion of the lake. Rounding it out with some of the benthic species, we've seen a similar story here with the sculpins, our native sculpin species, both slimy and deep water. Since really right after around 2000, we've tended to see much lower numbers of these fish. A couple increases here, but a lot of variability and generally lower numbers. Round goby, a lot of work being dedicated to exploring new methods to estimate the abundance of round goby in Lake Huron. Nonetheless, our trawl survey still catches this fish. We still estimate their abundance. We've seen generally a lot of variability. We know some of this is due to their abundance in certain types of habitats that we trawl or do not trawl. In 2020, we happened to pick up very low numbers of goby. So if we look at overall species composition, this kind of tells a more of a complete picture. And this is looking at the acoustic survey, again, 2004 through 2020 in the main basin. We can see in offshore waters, the dominant species in terms of biomass is bloater, roughly 80%. Rainbow smelt, roughly 8%. We see Cisco making up a little bit more contribution in 2020. But generally, this is really what constitutes the offshore prey fish community. The bottom trawl is a similar story. This is obviously the entire time series here, where we can look back and see more abundant alewife here in yellow. Rainbow smelt, much more abundant in the past. Bloater here. If we come into the more recent period here, the last 15 years or so, we've seen a few upticks, but generally it's been lower since around 2005, 2006. Lower and stable biomass, dominated by bloater. Again, nearly 80%, similar to acoustics in 2020. Rainbow smelt slightly higher, and then other species making up the rest. And there are some important factors to consider when we discuss the production of fish biomass in Lake Huron and some of the trends that we've been showing over the last number of years, particularly in light of some of the changes that have occurred in the long term over the last decade and a half, and generally reductions across the board in terms of prey fish biomass. We know that phosphorus is an essential nutrient for algae growth and a limiting nutrient in aquatic ecosystems, and that phosphorus is also positively related to fish biomass. Total phosphorus in Lake Huron declined by roughly 50% between 1995 and the early 2000s. While we have seen an uptick in phosphorus levels, it is currently stable but low. So generally, this decline in nutrients in Lake Huron means that less fish biomass can be produced. So sort of the trends and patterns that we're seeing in the last few years give us an indication that Lake Huron is sort of behaving like we might think it would in terms of nutrient limitation. When we think about a carrying capacity model for a population, just shown here with the population abundance over time, and the black line here just showing the sort of the limitation, the carrying capacity of that population. And just to point out, there's a lot of factors that can affect that, both density dependent factors and density independent factors as well. And we know that nutrients and how they play a role in aquatic ecosystems can certainly be one of these factors that can drive this down. If we look at the relationship between fish biomass and nutrients in general, we tend to see Lake Huron sort of in this portion of the curve where we have fairly low nutrients and low fish biomass. And just in conclusion, offshore preyfish biomass in 2020 continued to be dominated by rainbow smelt and bloater. Prey biomass has been stable for over a decade now. We have seen large reductions in the early 2000s, certainly around the time of the alewife crash, declines in other species like rainbow smelt. But the good news is it's sort of stabilized and has been at that point for several years now. Alewife biomass continues to be low. Native benthic species are also remaining at low abundance. We've been reporting on this for a number of years. Species like slimy and deepwater sculpin, native sticklebacks, these are historically important prey items. The native pelagic species, however, have increased and seem to have adapted well to this new food web, this nutrient-limited food web. So species like bloater, as well as cisco in the northern portions of the lake. And I'd just like to acknowledge all the USGS staff and Fish and Wildlife Service personnel who have contributed to the long-term surveys in 2020 and in previous years. Thank you very much for your time.
Wonderful. Thank you, Tim. Appreciate that. And there are a couple of questions coming in. If you have questions, even if we don't get to them now, type them in the chat and we can circle back at the end of the evening. But one question is, have there been any discussions or speculation on what the future of alewives and rainbow smelt might be in Lake Huron?
Yeah, thanks, Brandon. And thanks for that question. I guess I might need a little bit more information, but you know, when we think about prey fish, they're sort of managed indirectly by predators. And so when we manage predators, we're sort of indirectly managing the prey as well. It's sort of a secondary effect. There have been, I don't have the exact time frame, but over the last decade or so, there's been limitations on how many smelt can be harvested during the spring. So I think a recognition that the populations had declined enough to limit their harvest when they're most vulnerable in spawning streams. I guess as far as alewife go, I think currently the ecosystem is sort of, the lake is sort of not managed like it was, in the late 1960s, early 1970s, when we had a proliferation of alewife and rainbow smelt. Predators were certainly being stocked in higher numbers to sort of not only manage the explosion of exotic species like alewife, but also develop this fishery. There's sort of some of the original intentions for building the stocking program. So I think right now, I might defer to some of the managers as well on this, but the lake is not fully self-regulating. We still stock fish, but there's a little bit more in terms of self-regulation in the lake than there was, around the late 1960s, early 1970s.
Thank you. I'll grab a couple More questions here. One is in the model for energy transfer, there's no mention made of the introduced predatory zooplankton, like Bythotrephes, for example, the spiny water flea, inserted into the food web. Are these species being monitored and their effects modeled?
Yeah, that's a good question. So the food web was kind of a little bit oversimplified there, so it didn't really capture some of these other species. Speaking about, for instance, Bythotrephes, as Brandon mentioned, certainly one of our important predatory cladoceran zooplankton in the lakes, and non-native. There was some work done. I can't remember the exact publication year that looked at cladoceran or predatory cladoceran consumption of native zooplankton. Some work led by Bo Bonnell and others that came out of some work in 2007 that we did in Lake Huron. So there are studies that are ongoing that have looked at sort of what are the effects of sort of some of these predatory introduced zooplankton. And they are sort of being monitored, I guess, to answer that secondary question. We, on the acoustic surveys, we routinely sample both zooplankton and mysis, which are sampled with two different types of nets. And like Bythotrephes, for instance, are caught in both those types of gears. We do quantify their abundance in the zooplankton nets. They are a tricky organism. If you know about them, they tend to, the way that they're shaped, and they tend to clump up. And for the lab technician, they're a difficult animal to untangle literally in the lab, but we do nonetheless quantify their abundance.
Great, thank you. I will, there's a couple more questions. I'll throw one more at you and we'll move along with our evening. So two questions kind of grouping together here relating to the phosphorus reduction. So what's the primary cause of that 50% reduction in P in the lake, phosphorus in the lake? In Lake Huron, wasn't aware of the correlation between phosphorus and biomass. And so a secondary question was what's causing that low nutrient level in the lake?
Sure. First off, I think just going back, even predating sort of the mid 90s, we started, just across the lakes, there was tighter controls on nutrient releases into the lake. So the Clean Water Act, this goes back certainly further than the 1990s. And we began this process over time where we were, municipalities had tighter controls on how much nutrients could be flushed into the lake. That was certainly sort of began sort of the reductions in nutrients overall. We also know that invasive mussels, Dreissenid mussels, tend to, just by their very nature, filter nutrients. The way that they filter, essentially, that's how they're filter feeders. So they're filtering out nutrients, and that's also probably compounding that over time. So the spread of most recently the offshore variant of the Dreissenid, the quagga mussel, and more recently is expanding into deeper waters, is also sort of impacting that sort of continued, what we call oligotrophication, or nutrient reductions that are continuing. The water's very clear, as I'm sure most of you fishermen know that. As you go out there, you've seen the lakes much clearer. So there's a couple reasons. There's certainly lots of other causes for that we're not totally clear on in a lot of cases, but hopefully that helps a little bit.
Yeah, thanks, Tim. And I see, just acknowledging there's some questions about whitefish in a Cisco question, and maybe we'll try to circle back to those later in the evening. So with that, I'll a virtual round of applause for you, Tim. I appreciate your contribution. And I always kind of think about the importance of that prey base, as you said, Tim, as being the feeding factor to the great sport fishery that we all value and participate in. And so as an angler, we're often looking in stomachs and saying, gosh, what is my fish eating exactly? And so what I'd like to do is transition to our next talk with Jake Sawecki from the MSU Department of Fisheries and Wildlife. And they've been managing a long-term predator diet study that was actually started in part with USGS Great Lakes Science Center, looking at food web dynamics in Lake Michigan and Lake Huron through the lens or through the stomachs of our fish that we contribute as anglers. So Jake, welcome, and I'll introduce and pass the mic to you.
Thank you, Brandon. Hi, everybody. I'm Jake Sawicki. I am a PhD student at Michigan State on the Huron, Michigan Predator Diet Study. So I'm one of the people that has spent hundreds of hours in the lab digging through fish stomachs to see what these fish are eating. Before I get into it, I just want to mention that this is a very large scale project, and there are a lot of collaborators on this project. Without them, we wouldn't be able to get as many diets as we have collected. And some of those collaborators are some of the people that are here today. The volunteer anglers and sport fishing groups have donated stomachs to the project. And that has been really crucial for collecting data, especially in 2020 when a lot of our collaborators weren't able to get out and collect data themselves. So if you've been here before, you've probably seen these project goals, but for folks that haven't, the goal of the project, it started in 2017. We want to determine species-specific diets for salmon, trout, and walleye in Lake Michigan and Lake Huron. So we're looking at lake-wide diets of these different predator fish. And in doing so, we want to determine seasonal trends in diet composition. So looking at if these fish are eating different prey items at different times of the year. We want to look at spatial heterogeneity in diet composition. So are they eating different prey items in different locations of the lake? And then we're hoping to determine bias in angler caught diets. And what we mean by this is that the diets that we're looking at in stomachs from fish that were caught by anglers might be different than diets that are caught in surveys, just because the way that the fish are feeding when they're captured by an angler might be different than the way fish are feeding when they're caught in a gill net or a trawl survey. And then an additional aspect of this project is using stable isotopes to look at long-term diet. So over the course of a couple of months to see what these fish are eating rather than just looking at the stomachs and seeing what they ate within the past 24 hours. So far, progress has been good. We started in 2017. I joined the project in 2019, but due to the pandemic, we weren't able to collect as many diets in 2020 as we would have hoped to. We did collect some, but definitely not as many as previous years. And because of the pandemic, we've also been delayed in our ability to process stomachs quickly. So we did finish the 2019 stomachs in November of 2020 because we weren't able to get into the lab on campus for a couple of months. And we have all of these diets entered into an Access database. We have the results for the predator species for those stable isotopes. The prey fish have been submitted to the University of Wyoming, and they will get that data back to us, but we're still waiting on that. And another important note is that this project does end in December of this year. So this will be the last year that we are collecting fish stomachs for this project. And I'd love to see us collect more than we have in any of the years previously for this one final year of diet collection. So as I mentioned, we've collected a lot of stomachs thanks to the help of many collaborators. So here's some totals for Lake Huron and Lake Michigan. So the asterisk for 2020 is just because we're still processing those stomachs. So those numbers will increase. Unfortunately, in Lake Michigan, those numbers won't increase by much. We got very few stomachs from Lake Michigan this year, but in Lake Huron, we've done pretty well. So that's in part because there's many volunteers that have donated stomachs, and we were able to get to some fishing tournaments ourselves and collect some stomachs this year from Lake Huron. And for the first time since the project started, we're going to end up with more diets from Lake Huron than we do from Lake Michigan, which is exciting. So the diets that we've been collecting are from various locations in Lake Huron, and we do a pretty good job, especially in the northern part of Lake Huron, at getting diets from a variety of different species. As you can see in MH1 and MH2, we have tons of lake trout diets. We have Chinook salmon diets, even a decent number of walleye diets. However, in the other stat districts, so MH3 through MH6, we do struggle to get some samples from those areas, whether it's fewer people are fishing or there's just not as many fish to catch maybe. But if you are fishing in those areas and you do want to contribute to the diet study, we do appreciate getting stomachs from volunteers, and it would help us fill in these gaps so we can get that data that we need for those locations. We do well in MH4 for walleye. That includes Saginaw Bay. So you can see the 1600 walleye from MH4 there. In addition to spatial gaps, we have some temporal gaps. So people are fishing mostly during the spring and the summer, but during the winter and in the fall, as you can see, we don't get a ton of diets. And there's plenty of reasons for this. It's tough for us to go sample diets during these months, and there are fewer people fishing during the colder months of the year, so we just don't happen to get as many fish diets. So moving into some of the things that we found so far, and just so you're all oriented to how these graphs are going to look, on the y-axis of all these is proportion of diet, and that is by wet weight. So when we open up a fish stomach, we take out a prey item, we identify it, and then we weigh it. So that's what's on the y-axis there. And then on the x-axis on this graph, we have the different predator species. ATS is Atlantic salmon. Then we have Chinook salmon, coho salmon, lake trout, rainbow trout, and walleye. And the different colors represent different prey fish. So alewife are represented by that light blue. The dark blue is invertebrates. That really light green is other fish. So that could be shiners, gizzard shad, etc., which we just don't see a ton of. Another important one is that light orange color is round goby. Smelt is represented by the red, and the purple is yellow perch. So something that should jump out right away is that the diets of these different predator fish are pretty diverse in Lake Huron, but they're pretty consistent from year to year. So if we look at Chinook salmon, for example, that's that second bar from the left, they eat a good number of alewife and rainbow smelts in Lake Huron. Walleye, which are far to the right, eat a lot of round goby, represented by that light orange. And then, as we're seeing in 2019 and 2020, they're starting to incorporate more yellow perch into their diet. However, in 2020, this could be because our samples came from later on in the year, whereas we see round goby in the diet more so in the spring. Then if we look at lake trout, we see that they incorporate a lot of round goby into their diet, which I don't think is a surprise. And there's also a decent number of rainbow smelts in their diet, especially in 2018 and 2020. And then for rainbow trout or steelhead, they eat a lot of bugs. So that's that dark blue color. So if we look a little bit more closely just at some of the main predator fish in Lake Huron, what we see for Chinook salmon is that they're eating more alewife over time. So if we look from 2017 to 2019, the proportion of the diet made up by alewife was increasing. However, we don't see that same thing in 2020, but that's because we only had 38 fish, maybe. We probably need a bigger sample size to be able to accurately see if that trend was going to continue or not. If we look at lake trout, in any given year of the study, round goby have made up anywhere from 30 to 80% of their diet. But we also see that rainbow trout have a strong presence in the diet of lake trout. So rainbow smelt in 2018 made up about half of the diet, as well as in 2020. And then for walleye, round goby are an important prey item for walleye. Yellow perch look like they're becoming more of a common prey item. However, that 2020 sample, most of it comes from Saginaw Bay, and most of it comes from later on in the year when the walleye are maybe eating the young of year yellow perch, because we do find small yellow perch in the diets. And we may be underrepresenting the round goby there just because we don't have any diets from the spring for those walleye. And then looking at steelhead, they feed almost exclusively on inverts, so they eat a ton of insects. There's some alewife consumption that is going on or that occurred in 2020. So they did switch to something a little bit more substantial than just consuming inverts in 2020. And then if we look across time, so starting with Chinook salmon, if you look at the left graph first, this combines all years of sampling. We see that alewife were pretty common in the diets in the spring. And then as the season progresses, they seem to incorporate more rainbow smelts into their diets. We see that occurring pretty strongly in 2017, a little bit in 2018. We don't see it as much for 2019 and 2020. If we look at lake trout eats a lot of round goby in the spring. That's represented by that light orange color. And then as the season progresses, they look like they switch to rainbow smelt. This is particularly noticeable in 2018 and 2020, where we saw that the diet of these lake trout was almost 50% rainbow smelt or more. And then in 2019 and 2017, we see this a little bit, but we do see that round goby remain more important later on in the season than they do in 2018 and 2020. And then for walleye, so I did mention that walleye were consuming more round goby in the spring. We can see that here. So in June, they're consuming a ton of round goby. Same with April. And then as the season progresses, so we get to July through October, they're starting to incorporate more yellow perch, which again, we do see very small yellow perch in the diets of the walleye. So it could be that they're eating those young of year yellow perch that they can't eat in the spring, but they can eat later on in the season. And then lastly, if we look at steelhead, we can see that they like to eat a lot of inverts in the spring. So that is that dark blue color. And then they make the switch to smelt and alewife later on in the season as you get to August, September. We see in 2017 a lot of rainbow smelts, 2018 a lot of rainbow smelt, and 2019 a lot of rainbow smelt. But in 2020, they seem to be consuming alewife more frequently later on in the year than they are rainbow smelt, which is interesting because we see a difference for lake trout, which are actually consuming more rainbow smelt later on in the year after they switch from the round goby for 2020. So the next steps of this project are to continue analyzing the 2020 stomachs. So we do have plans to finish that this summer. Like I said, we didn't get as many as we would have hoped for, but that's okay. We're going to be able to process those by the end of the summer. We have the isotope data coming in, just waiting on the prey species, and then we'll be able to analyze that and have those results available. We're going to start collecting more stomachs starting this month, actually. I am going to a fishing tournament in Port Huron on Saturday. So if you're there and you want to donate stomachs, I'll be there to collect them. And then the project, as I mentioned, also ends in 2021. So at the end of this year, we'll be done collecting data, and then we'll just be processing those stomachs and analyzing the data. And if you are interested in participating in the study and you want to volunteer and donate stomachs, we are introducing punch cards for 2021. So this is what they look like. They're going to be one full sheet of paper. I had to cut it in half for the PowerPoint so it would fit. But if you're interested in receiving some of these so that you can collect stomachs, let me know. We'd be happy to send them to you directly. If you want to have some to put at your local fish cleaning station, let me know that too. We can send you a packet, get you all the materials that you need so that you're able to put these out, and then people can help collect fish stomachs and contribute to this project. And with that, if you want to stay up to date on the project, we do have a social media page, the Huron Michigan Predator Diet Study. You can follow us on there. You can follow me directly on Twitter at Jake Sawicki. I tweet a lot of pictures of fish diets, so round goby, alewife, and rainbow smelt that come out of fish stomachs. And you can email me at my email address there. Thank you very much, and I'd be happy to answer any questions.
Thank you, Jake. And just a note for everybody, we are dropping and sharing some of the website and resource links in the chat, so you can grab them today, and we'll make sure to share those in post-workshop communication also. So this is definitely a study that's dependent on angler contributions, so we're hoping everybody and all either are contributing or will contribute this season. So Jake, we do have one question relating to the calorie value of prey species. So there's a large difference in the calorie value of the different species you're finding in the stomachs. Have you looked at the actual energetic content of these prey species for each of the sport fish diets?
For this project, we have not done that. I know there is research out there where that has been done, because there definitely is a difference between the caloric value of certain prey items, but for us personally with this project, we have not done that. No, it's definitely something that we could do though.
Great, thank you. Are there any specific seasons or species that you could use a little extra help, from anglers with?
Definitely early on in the spring, we don't get a ton of samples. In the winter, of course, although that's difficult, and then later on in the fall. So seasonally, late fall or beginning of fall even, and early spring. In the summer, we do pretty well. Species wise, we do really well for walleye from Saginaw Bay. We do well on lake trout, but rainbow trout, we're always looking for rainbow trout. Any of the salmon, that'd be valuable. We don't get a ton of those. Brown trout, we have very few brown trout.
Great, thank you. I appreciate that. And I think it's worth noting that you guys spend a lot of effort this year trying to make this an easier project for anglers to participate in, trying to streamline the contribution piece. We do have one more question that just came in. Is there any difference in preference within a given game fish species for a particular prey other than perhaps by size, or is it more happenstance or prey availability?
There is definitely preference by some species for certain prey. I didn't show any of the Lake Michigan data, but Chinook salmon will almost exclusively feed on alewife when alewife are available. Lake trout are more of a generalist. They'll not be as picky, but for a species like Chinook salmon, they are very picky and they want to eat almost exclusively alewife. Whereas fish like lake trout, less picky, coho salmon, less picky as well, but there is some preference involved based on predator fish. Great,
thanks, Jake. Appreciate that. So a virtual round of applause and much appreciation for sharing that study. And I think to keep us moving on with our program, I'll move us up the food chain a little bit. One of the traditional updates that the DNR Fisheries Research Station provides is an offshore fishery update, catch and creel, sort of look at how the fishery has performed and some of the highlights that fishery provided in the last year, and maybe some things we can look forward to in the coming years. So Todd Wills is not able to join us tonight, but he pre-recorded this review and update for us to listen to, and then some of the DNR team is on hand to answer any questions you might have following Todd's update.
Good evening. Thanks for tuning into tonight's Spring Fishery Workshop. Before I begin tonight's update on Lake Huron's main basin offshore fishery, I think it's helpful to define exactly what a fishery is. A fishery is the interaction between fish, habitat, and people, which in the case of the Great Lakes and its connecting waters results in major social and economic benefits for Michigan. When we speak of a fishery and fisheries management, we're talking about these three things collectively. Tonight's talk will tell the story of the main basin fishery through a combination of data from our charter reporting and angler survey or creel programs. I also think it's helpful to begin by providing background on the changes that have occurred in Lake Huron in the 1990s and 2000s to set the stage and provide greater context for what we've observed in the main basin fishery. This 50,000-foot level overview of how the current conditions in Lake Huron came to be begins with the introduction of invasive zebra and quagga mussels. This created a perfect storm, which led to a decrease in offshore prey through fundamental changes in nutrient and energy flow, which was exacerbated by increased predation pressure from naturally reproducing Chinook salmon. We're all very familiar with the results of these broad ecosystem changes, like the disappearance of alewives in the mid-2000s and the resulting crash of the Chinook salmon population. The fishery has indeed changed too, but it's thriving with a resurgence of native species like lake trout and walleye, with many other species that add diversity to the catch. Let's look first at the data from our charter fishery and a little background on how we collect this information. But before that, let's also acknowledge that 2020 was a very different year because of the pandemic. Keep that in mind, as it not only affected our ability to collect and summarize data, but it also created a surge in interest for outdoor recreational activities, including fishing. Charter operators are required to report their effort, catch, harvest, and release to us each year. We don't survey our charter operators. Instead, we compile annual summaries of the data that they report to us. Let's begin with a summary of charter effort. Main basin charter fishing effort during 2020, over 23,000 angler hours, continue to trend above the contemporary average of over 22,000 angler hours, which is represented by the black line. This is the 5th consecutive year of above average charter effort, and that's impressive considering that charter operations were undoubtedly affected by restrictions associated with the pandemic, especially early in the year. Note that the changes to the Lake Huron ecosystem that have occurred since 2005 can be considered the new normal, so it's more representative to focus on trends during the last 15 years, which I'll continue to do throughout this talk. I use harvest as one of the primary measures of fishery performance throughout this talk, since the main basin fishery is harvest-oriented, with the exception of tributary fisheries for steelhead and Atlantic salmon. Harvest in the main basin charter fishery mirrors total charter effort, which is represented by the black line. Total harvest in 2020 was just over 11,500 fish, which is a slight increase compared to 2019. Salmon and trout continue to comprise the largest component of charter harvest, followed by walleye. Yellow perch have been a relatively minor component of charter harvest. Note that the salmon and trout category includes Atlantic salmon and pink salmon for the first time in 2019. In contrast to the charter reporting program, the Creel program is a survey. We station Creel clerks at ports in order to interview non-charter recreational anglers as they finish their fishing trips. The clerks are also responsible to count the number of people or boats that are fishing at scheduled times. Counts are like taking a snapshot of fishing at a given place and time. Counts can also be done by plane. Using the interviews and the counts, the Creel program estimates how many hours people spend fishing, how many and what species of fish they harvested and released, and what the catch, harvest, and release rates are for each species from year to year. We estimate this information because we only sample a proportion of the actual anglers that are out there fishing. 2020 was a very different year for Creel. The annual Creel survey started nearly 2 months later than usual because of the pandemic. So in a typical year, the open water Creel covers the months of April through October. In 2020, the Creel covers the last week of May through October. Please remember that difference when considering comparisons among years in the next slides. First, here are some big picture data to address the question, where does Lake Huron fit in relative to the other Great Lakes? Lake Huron, the main basin and Saginaw Bay combined, accounted for more than a third of Michigan's total Great Lakes angling effort in 2020. This was more than the effort in Michigan waters of Lake Erie and Lake Superior combined, and is nearly equal to Lake Michigan. Note Lake St. Clair is excluded from these calculations, as the 2020 Creel survey there was shorter than the other Great Lakes. In a similar comparison of total catch, Lake Huron, the main basin in Saginaw Bay, accounted for over a third of the total catch, following Lake Erie and exceeding both Lakes Michigan and Lake Superior. Let's focus in on the main basin fishery for the remainder of this presentation, since this is an offshore fishery talk. Only main basin ports are included for the following summary slides. The St. Mary's River and Saginaw Bay are not. Data are pooled across all sites sampled in a given year to provide the big picture. Estimated recreational fishing effort during 2020 ranged from a low of about 400 angler hours at Hammond Bay to a high of nearly 80,000 angler hours near the Lachino Islands. Drummond Island and Rogers City are also high-effort ports in the north, while Alpena and Oscoda received the highest effort mid-lake. Lexington followed Harbor Beach and Port Sanilac in the south. Overall recreational fishing effort has trended down since 2005, but a declining trend in recreational fishing effort isn't unique to Lake Huron. Main basin total recreational effort during 2020, nearly 310,000 angler hours, was about 30,000 hours higher than 2019. And remember, that's only for part of the year. While still below the contemporary average represented by the black line, the past 3 years have shown relatively stable levels of effort. Salmon and trout were the major sources of non-charter recreational targeted effort throughout the 1990s and early 2000s. After the food web changes in the mid-2000s, more effort in the main basin has shifted to walleye and yellow perch. Targeted salmon and trout effort in 2020 was similar to 2019. The increase in non-charter effort in 2020 was due to more walleye and yellow perch effort. It doesn't appear that the observed decline in non-charter recreational fishing effort in the main basin is due to a shift in effort to Saginaw Bay. While lake-wide effort, represented by the blue line, has declined, Saginaw Bay has consistently accounted for 60 to 80% of lake-wide effort over the past 15 years, which is represented by the red line. The proportion of angling effort directed to Saginaw Bay did increase around 2004-2005, when the major change in the Lake Huron ecosystem occurred. Similar to the charter fishery, non-charter recreational salmon and trout harvest closely mirrors main basin targeted effort, which is represented by the black line. Chinook salmon, lake trout, and rainbow trout are important species to the fishery and have historically comprised the largest component of the trout and salmon harvest and continue to do so. Non-charter salmon and trout total harvest during 2020 was just over 26,000 fish. That's right at the average for the time series. And remember, it's an underestimate because the Creel survey didn't cover the typical April through October period. I'd like to focus in on Atlantic salmon for a moment, as they weren't included in the previous summary slide. Atlantic salmon harvest has been variable during the past 15 years. For reference, I've included the number of fish stocked, which is represented by the black line. The harvest in 2020 was about 900 fish. The fishery continues to grow in popularity beyond Lake Huron, with catches reported throughout the St. Clair-Detroit River system. The extent of the Atlantic salmon fishery is not fully captured by the Creel survey. By all accounts, there are active tributary fisheries, such as the AuSable River and Thunder Bay River, as well as fishing activity that extends into late fall, which is confirmed by coded wire tag reports. Note that catch and release is also not reflected. A notable highlight for 2020 was rainbow trout harvest, which more than tripled when compared to 2018 and 2019, and was more than double 2017. Total rainbow trout harvest, about 4,200 fish, was the highest observed since 2012. Harvest rates are another useful measure for fishery performance because they account for effort rather than just raw numbers of fish. Overall non-target harvest rates, or the number of fish harvested per angler hour of effort for salmon and trout, have been above the contemporary average, represented by the black line, during the past 5 years. The salmon and trout harvest rate in 2020 was the third highest in the time series, following 2018 and 2016. Let's shift attention away from salmon and trout for a moment and discuss walleye, which have responded very well to the ecosystem changes in Lake Huron. The recovery in Saginaw Bay is a perfect example. If you're interested in learning more, be sure to tune into the Nearshore Fishery Workshop on April 29th. Main Basin walleye harvest in 2020 was around 7,000 fish, which is on par with the past 2 years. Remember that effort drives harvest, and targeted walleye effort in the Main Basin, represented by the black line, has been highly variable. I'd like to close the individual data summaries by touching on yellow perch. Yellow perch harvest was very low in 2018 and 2019, with 2019 effort being the lowest in the time series. Effort increased in 2020, and accordingly harvest did too, with an estimate of just over 85,000 fish, which was double that of 2018 and 2019. In summary, charter effort and harvest have been stable and above average during the past 5 years. Harvest mirrors effort. The non-charter recreational fishery has been relatively stable the past 3 years, with a small increase in effort and harvest for 2020. Lake trout remain the largest part of the harvest, and overall salmon and trout harvest rates are up. The anglers that are investing the time to fish are being rewarded. Finally, Lake Huron continues to support a diverse fishery. Anglers continue to harvest lake trout, Chinook salmon, rainbow trout, Atlantic salmon, and coho salmon. The relatively new coho salmon stocking program initiated in 2019 will create more opportunity. And don't forget about the opportunities provided by walleye, as well as other species like yellow perch and smallmouth bass, which are not mentioned here, when fishing the Main Basin. Thank you.
And thank you goes back to Todd for preparing that update. So, and I'm going to move us on and introduce Pete Esselman with the USGS Great Lakes Science Center. The Great Lakes Science Center has been doing some pretty innovative work to really use some technology to think about how to monitor and estimate round goby abundance. We all know gobies end up in the stomachs of a lot of the fish we're catching, but what is that overall contribution of gobies in our Lake Huron system? So Pete, I'll introduce you and let you share a bit more about your exciting work.
Thanks so much. I appreciate it. I appreciate the opportunity to present tonight. Out of the gate here, I want to acknowledge my collaborator, Chuck Medengen, also at the USGS Great Lakes Science Center in Ann Arbor. Today, I'm really pleased to be able to share some essentially work with emerging technologies to address some pretty important gaps in our capacity at USGS. As you heard from some of the other presenters earlier, Jake and Tim, for instance, round goby are a very important prey item for lake trout, walleye, as well as other species in Lake Huron and the Great Lakes more generally, especially the lower four lakes. And through the effort that USGS expends every year with the bottom trawl in particular, we've come to recognize that during the season when we're doing our sampling, the gobies tend to be distributed from 0 meters depth to about 30 meters depth, and most of our bottom trawling effort happens outside of that. We're fishing from large vessels. Those vessels don't go very shallow very easily. We're fishing with bottom trawls that may skip over the gobies. The gobies may have behaviors that would make them avoid the net. And so there's the assumption that our estimates of round goby have been consistently low. In other words, we're not very efficient at catching them. And at the same time, we're seeing these great increases in goby as in the guts of the different fishes that are important within the sports and commercial fisheries. And so we've essentially begun developing new methods. And the method we've developed is one that relies on what's called an autonomous underwater vehicle. And it's essentially an underwater drone that we've equipped with cameras. You can see the vehicle in the lower right side of this slide here. So this is about 9 feet long. It is untethered. Essentially we plot out a mission for it. We load that mission via Wi-Fi link to the computer on board the vehicle. We deploy it from a small boat, which I'll show you in a moment in a video. It operates, we tend to operate at about 1.5 knots at 1 to 2 meters off the bottom. We tend to target 1.75 meters off the bottom. And the vehicle essentially has been modified to carry a color camera, which is a 9 megapixel camera, and then we have a stereo camera. Of course, the stereo camera gives us the ability to do 3-dimensional reconstructions of the lake bed, so we can understand not just where the gobies are at, but also we can understand what type of habitat they're existing in. The vehicle is rated to 200 meters depth, so we can essentially capture the entire summer and potentially winter range around Gobi. Importantly, we have a very sophisticated positioning system. It's essentially it's called a fiber optic gyroscope. A gyroscope essentially can detect angular acceleration due to currents or upwelling or density changes in the water. And this particular vehicle accounts for that, so that when we lose our GPS signal at the surface, it maintains a sort of a model of where it is based on any sort of angular acceleration that it's subjected to. So you can see in this video what it looks like in the water. This is in Thunder Bay, Lake Huron, after a deployment. We just hook it up. We bring it on the back deck. This is before COVID, when we had to have our masks on all the time. And when it's underwater, it's essentially flying right off the bottom. You can see this is probably about 6 feet off the bottom. There's an object avoidance sounder in the front, which is essentially if there's a shipwreck or a pipeline or anything in front of it, it will avoid that. The orange mast is essentially a communications mast when we're at the surface. And then it's taking photographs along the way. And we take 5 photographs per second as we move. So this is what the pictures look like. You can see, if you can recognize some fish in this picture, also some hairy cladophora algae. Those fish can be labeled or traced manually, but we're also trying to train machines to do that. This is essentially what the process looks like of from a raw photo to having data from each photo. So in the top left, we have the raw photo. Right now, we have hired a company in New Jersey to look at each individual photo and to trace the fish that are present in that photo. Once we have those traces done, we can actually estimate their length of each individual fish, which gives us the ability to estimate their weight as well, because weight scales with length. And once we have the whole photo sort of labeled and quantified, we can get the numeric abundance as well as the weight of each fish, and then we can calculate the density, which is the abundance per unit area, in this case meters squared, using the field of view of the camera, which gives us a known area on the bottom. So when we average our data over many images on a transect, usually we subsample the images so we're not getting double counts, and then we could average them over many transects within a lake. And that's exactly what we are working on right now with 2020 data we collected in Lake Michigan. So last summer, we overcame the COVID barriers, and we were able to collect 30 10-kilometer flight paths with the AUV. We tried to reach that blue line, which is the 30-meter depth line in Lake Michigan. We structured our study to encompass coarse bottom types, rocky bottoms, which would be the red dots, as well as fine bottom types, which would be sand and silt and clay. And so, we were able to essentially have a very nice spatially distributed study that was focused on the summer distribution of goby, which is in the shallow water environment from 0 to 30 meters. For much of the rest of the talk, I'll be focused on this site, LM 20_08, which is to the west of Grand Traverse Bay. You can see it outlined here. So the first thing that we did, well, in total, we gathered about 2.5 million photographs during this effort. And that's just from the color camera. And we have the same amount for the stereo camera. And we have point clouds, which show us the elevation of the lake bed. It's essentially how rough it is or how smooth it is. So we subsampled every 25th image, and then we screened all of those, and we threw out any pictures that were overexposed, underexposed, too turbid. And we had a fairly consistent overexposure in our photos, which sort of begs for some camera adjustments this year. So we ended up being able to use about 44% of the 100,000 photos. And for those usable images, we sent them off to be evaluated and traced for the presence of fish. You can see Sleeping Bear Dunes shown in the pink outline here. Sleeping Bear Dunes has this heart-shaped reef called Good Harbor Reef. And this is showing the elevation of the reef, with the warmer colors being shallower and the bluer colors being deeper. So last August, we were out there. We did a 10.5 kilometer flight path with the AUV, lasting almost 4 hours. We encompassed 3 meters to 26 meters depth. And we were able to gather about 88% of our images, 3,000 images were usable. 25% of those had fish. And from those, we can then take the traces. We've developed an automated approach to fit a midline, the red line in the left images of these fish. And that gives us the total length of each fish, which then we can convert to the grams that each fish weighs based on some standard curves that we have. The plots in the middle and on the right essentially are showing that the higher the bar, the greater number of fish that fell in a certain length category. And so on the horizontal axis at the bottom, it shows different bins of 10 millimeters in length. And you can see that most of the fish were in the neighborhood of about 70 millimeters. And we can also further break that down into depth bins. So on the right, you can see in the 10 to 15 meter bin, we had the greatest abundance of round goby on this particular survey. And we can, of course, get statistical estimates of the number of fish per meter squared, as well as the weight of fish per meter squared based on that length to weight conversion that I mentioned. And so when in my title, I suggested that we're trying to make a whole lake estimate of round goby to complement our trawl-based studies. And the way we do that is essentially through extrapolation, a mathematical extrapolation, and I'll demonstrate that briefly here. So here we have Good Harbor Reef in its depth ranges. We can essentially categorize that depth gradient into discrete depth classes, and each one of those has an area associated with it, which you can see on the left-hand side of the slide. Now, I already showed you that we can also calculate numeric and biomass density within each bin. And then if you multiply area by the number of fish per area or the grams of fish per area, we can essentially come up with the total number of fish estimated in each depth bin, as well as the total weight of fish. And I did make a little conversion from grams to kilograms on the biomass estimate here. So in this case, in this 10 square kilometer area of reef in northeastern Lake Michigan, we estimate there's a little bit over 7 million fish on the reef, and there's weighing 89,000 kilograms, which is 0.089 kilotons. So that's just an example from one of the 30 transects we did. So we're in the process now of combining those. But even looking at that specific transect, we can learn some things. So first, we can learn that the abundances that we measured are sort of well within the range of other published studies that used similar methods using video or cameras to estimate fish abundances. The other thing that we can learn is that even in a 10 square kilometer area of Lake Michigan, which is a lake in which we through our bottom trawl survey, make an estimate of fish abundance in 15,000 square kilometers. Even in that small 10 square kilometer area, we're already estimating enough round goby biomass to account for 3% of the average lake-wide estimate that we get from our bottom trawl, suggesting that we're likely to arrive at an answer eventually at the end of the lake-wide sort of data crunch that is a fair amount higher than our bottom trawl estimates coming out of the large vessel trawling operations. These numbers are likely to be conservative because round gobies, of course, are cryptic. They're well hidden. They hide. They're under the algae. They're under the rocks. They bury themselves in sand sometimes. And so, we're likely to be estimating on the low end of abundance when we come out with our numbers. And of course, we're currently working to combine all these data to get to the Lake Michigan scale from this sort of transect scale that I just showed you. The other thing I wanted to sort of highlight very briefly is that I mentioned that we're trying to incorporate not just these depth categories, but also whether the substrate on the lake bed is smooth or rough. With the assumption that rougher substrate correlates to greater abundances of round goby, which is a well-established fact in the literature from the Great Lakes. So here's an example of a three-dimensional reconstruction that's coming from our cameras. This is a reconstruction that is probably has a resolution of about 1 centimeter, maybe a half centimeter. You can see these the clay. This is actually an area on Mitchley Reef from Thunder Bay, Lake Huron, and there's these clay platforms there, which are shown in smooth in the reconstruction. And there's also these Dreissenid mussel covered rocks, these quagga mussels coating these rocks. So we can correlate this sort of high resolution habitat information with the abundance of gobies and improve our estimates of abundance. And we're also working to sort of take the middleman, the people who are labeling the fish, we're working to take them out of the equation here. So specifically, we're really trying to automate the process of detecting the fish in the imagery. There's very powerful approaches using AI systems. You can see in this case that a functioning model that we're calling GobiNet has detected with between 87% and 99% probability, which is shown here as a proportion. 0.87 would say there's an 87% probability that round goby occurs in that bounding box. So we're basically, we already have the computer performing at about 82% accuracy in automating that. And we're moving forward. So we're going to incorporate coarse versus fine substrate as well as depth classes into the whole lake assessment. Hopefully within 2 months, we have our final results for that and we can share those and publish them. We are essentially expanding our program, building a second autonomous underwater vehicle. That should be online in the early fall. We're also redesigning our camera systems with the delivery of around the same time to have better exposure, better lighting. And we're importantly expanding to Lake Huron this summer. And so currently it looks like Canada might be off the table for us, but much like last year's acoustic survey and bottom trawl survey, we'll likely be working on the US side of Lake Huron at multiple sites. So hopefully I run into some of you this summer. And if I do, please feel free to come up and check out the robot and talk to me more about it. And we're very much pushing to develop these automated methods more quickly, because the vision here is that by the time the robot comes out of the water, we take it, we put it in a box trailer, we upload the data to the cloud, and the computers are essentially whirring away remotely. And within a few days, we can publish the results of a specific transect. And anyone could go there and sort of consume that information and the maps that we're predicting goby abundance variation in space and in time. So with that, a lot of folks, a lot of funding has supported this project. Some people on the phone today, Randy Claremont, thank you so much. A lot of people on my team listed on the left. And if these can be shared or you can consume this video later, these are some of the references that I shared in the presentation today. Thank you very much.
Thank you, Pete, this is all very exciting, and I appreciate you taking the time to share a summary of the technology and some of the early findings and future opportunities. That's really, really neat. So there's some questions coming in. One question I had was, how does the image, how are you differentiating between other bottom dwelling species like sculpins and darters that look kind of similar to, or could look similar
Right.
to?
So we're working, so currently I think the task is to automate the process of identifying a fish on the lake bed, regardless of species. My experience, I'm one of the scientific divers at USGS, and when I'm down on the lake bed, the vast majority of fish in the shallow zone in the summer, that would be sort of relatively stationary and able to be photographed, are round goby. We have trained technicians that are discriminating between slimy sculpins and deep water sculpins and round gobies and some of the other benthic fish that we might encounter. So I think it's fair to say that our estimate is, is maybe more accurately characterized as being one that for benthic fish that are cryptic and that don't move a lot. But I think it's also safe to say that the majority of those fish are round goby. And so I'll leave it at that. I think down the line, as our image quality improves, we'll be able to discriminate among species. I don't think we'll do that this year or next, though.
Yeah, thanks, Pete. And then one other question I had here. Regarding the potential to deploy these types of underwater ORVs remotely to get a more comprehensive picture of prey fish in the lakes, in really specifically in the context of supplementing or maybe replacing some of the trawl data. And I know, so Tim and you work together. So Tim was sharing some of the acoustic data in the trawl data, and how does that pair up with the data that you're gathering in terms of telling overall prey story for Lake Huron or
Sure.
Lake Michigan?
Sure. So the large vessel program is kind of the heart of USGS's science, and the thing that's really powerful about our data set is that the bottom trawl data set extends now for 50 years, and we wouldn't have as much insight into these food web shifts that Todd was talking about unless we were out there every year deploying the similar gear types with similar boats to get comparable data. As we move to new technologies, the opportunity is to assess certain species better that we know we're missing. And it's also to fill in gaps, because when we're out there with a large vessel, there's a lot of effort. We can't be everywhere all the time. And if the weather gets rough, we can't be on the lake at the shoulders of the seasons when whitefish and cisco and lake trout are beginning to spawn and deposit eggs and aggregate. We can't be out there very effectively because it becomes pretty dangerous. And so these technologies we see as a supplement and a complement to our large vessel operations. I think it is worth making a comparison and trying to sort of cross-calibrate between the new tech and the old tech. But I think right now the mode of thinking is not that these new technologies replace the large vessel operations and the important data we get from those operations, but rather to supplement those.
Thank you. And then there's one last question here in the Q&A tabs. Have you repeated transects as a means of data controls? So it sounds like a lot of the work right now is calibrating and test driving the equipment and the methodology. And so just wondering about it, how you're managing for data controls.
Sure. Not exactly sure what the question, what data controls means in that context. What we have been doing, we do repeat transects, and because we have this underwater positioning capability, those transects are likely to be within a few meters of each other, regardless of the year or the month or the day or the time of day. And so we can do a pretty good job repeating the same transect over and over. What we've been doing so far is we've been doing night-day comparisons. And the reason we're doing that is because, it's possible that we could more effectively detect round goby or other species at night than we can in the day, especially in the shallow water environment. And so we're trying to understand our catch efficiency through repeated sampling, including sampling at night. I think there's also, I think it remains to be seen a little bit whether we run the exact same transects every year. But we certainly could do that. And so we could control for year to year variation in the habitats we assess by essentially running the exact same transects that we did last year over again. We could probably even get pretty close in terms of when we assess those transects. So that would be one form of control. But there's a trade-off, of course, when you start talking about, looking at the same 30 sites every year versus potentially branching out and learning more about the spatial variability, like essentially how variable are those abundances in space. For instance, we encountered massive aggregations of gobies, I would say 75 gobies per square meter near Ludington last season in early September. And it's possible that if we can continue to sort of mix up where we're doing our transects, we could encounter additional we can make new realizations about sort of where gobies are and why they're there and when are they there. Speaking about when, there's the common thinking right now based on available evidence is that gobies go to deep water in the wintertime. And that's the other thing that we can do pretty well with this, because we can get sort of a surgical precision of our deployment well down into the lake water column to 200 meters. And so this offers us a great opportunity. For instance, if we were going to, again, do repeated transects, one way of doing a repeated transect that would be particularly useful is to do that on a monthly basis or every other month throughout the year to see if the distribution and sizes of the gobies we catch are actually becoming deeper in the winter and then coming back into the shallows, to confirm that pattern for Lake Huron, because it hasn't yet been confirmed for Lake Huron in a sort of at least in the scientific literature.
Thanks, Pete. And again, a virtual round of applause and thank you for taking time out of your evening to share with us. It's all very exciting work, and I'm sure folks on the webinar are excited to have had a chance to listen in. So thank you. And I appreciate everybody hanging tight. We've got a great group tonight, and everybody's hanging strong into the conversation. So we're getting close to rounding out the evening, and in good tradition, we end our workshop series with each year with a fisheries management so what, right? So we've had a great opportunity here from a lot of the research side of what's going on in the Lake Huron fishery. And we're going to turn it over now to Randy Claremont and his team, Dave Borgeson and Jeff Jolly, and some other of the management crew are online to really share a quick management update, but maybe more so generate a conversation. And I did just want to acknowledge that there was some questions in chat regarding comparing and contrasting Atlantic versus coho stocking. And I think that probably relates here, right, Randy?
Absolutely, Brandon. And just want to say thanks again before I forget to you and Michigan Sea Grant for all your help in facilitating these, and to the presenters for the excellent presentations and information they shared. We all know that 2020 was a challenging year. And, as we saw in some of the estimates that Todd Wills provided for the charter and recreational fishery, there's gaps in data, but crews did their best and staff did a phenomenal job trying to complete the surveys or get whatever information we could to inform our management decisions. So kudos to all of the staff and everybody for, really doing their best in a challenging year. one of the things that we looked at that I want to talk about before we get into maybe the coho and the Atlantic salmon is, we did start the year with a lot of rapid changes, and one of those where we had to drop our spring egg takes because of the pandemic. So no muskie, no walleye, no steelhead egg takes occurred. That impacted our fish stocking numbers going forward. But one of the things that we did is we tried to use those gaps as opportunities to look at our programs and kind of do a deep dive into some of the approaches. And before I get into that for steelhead and Atlantic salmon, I want to talk about input from the advisors. And I see, with the 100 or so participants we have on today, that many of our Citizens Fisheries advisors are on the call and are very much familiar with giving us input throughout the year on various proposals. One of those being, their concern with the post-stocking survival of, especially for steelhead, but also Atlantics and really for most of our stockings in general. But there were specific cases that our advisors raised that they were looking to see if there are any other options to increase the post-stocking survival of some of our salmon and trout. And because we weren't stocking steelhead and walleye, we kind of took the opportunity to work with our advisors, and we came up with what I believe is a relatively flexible but advanced strategy that we're going to implement next year to try to deal with the timing of these stockings and increase the overall probability of post-stocking survival so that these fish return to the fishery. And we did that for steelhead and walleye, or steelhead and Atlantic salmon, presented that to the advisors, and that was well received. Likewise, we did a similar type of exercise for walleye in our pond production. This is mostly for inland systems, but again, trying to utilize the opportunity that we had because we weren't stocking fish to try to address some of our management concerns. So, I could go on with updates, but I really think if there are some questions out there, I'd like to hear them. I know one of them that was asked was, the advisors worked with us on some revisions to our salmon and trout management plan a few years back, and that resulted in restocking coho salmon. We discontinued coho salmon stocking Lake Huron in 1989, but we just recently reinstated it, and part of it was to deal with the changes in the prey fish community and the nutrient levels that Tim talked about, that Pete talked about, and that coho would be a better fit. But we didn't want to increase the overall predator consumptive demand. So we decreased Chinook salmon at roughly the same equivalency in terms of their overall consumption, at a rate that would allow us to increase or to reinstate coho salmon. So we traded Chinook, 30,000 Chinook for approximately 90,000 coho. And anglers are seeing those fish return to the fishery, especially in the southern portion of the lake in the spring. So really great news. We'd love to hear those reports. But some of the points where, well, could we stock more coho, or what other changes might be coming? And I really want to say, we looked at our overall predator-prey dynamic and made some assessment that we really don't want to push the system much more than we are right now. And there was an opportunity with the reduction of, no steelhead eggs being taken last year, that maybe we could accommodate or temporarily increase salmon stocking of a different species. And our advisors said they would rather not do that in lieu of the prey fish biomass levels, and give a little bit more room in the system to manage a healthy ecosystem. So that's a mouthful, I know, and it's a lot that we have been trying to work through. But with that, maybe I can, transition into some questions. It looks like a comment that the coho and the Atlantics are a good part of the fishery. Are we getting data about the Lake Erie corridor? So we are getting reports of coho showing up not only in the Lake Erie corridor, but also on the Ontario side of Lake Huron. So the coho, that's telling us they're surviving well. So we would see, you know, the range expansion if we get good survival, which appears to be the case. So to us, it's a good indicator that these fish are surviving well, and we expect them to kind of migrate north as the season warms, and we see that in some of the creel data. So we'll expect that. What are we hearing on coho fishing this spring, the south end of the lake? Are many being caught? Kind of address that, but basically, yeah, I would say the fishery continues to expand. Last year in 2020 was the first spring we were expecting returns, and unfortunately, we had to postpone our creel surveys. But a lot of you sent me images of fish that were being caught, limit catches of coho in southern Lake Huron. Similar pattern this year. So we're seeing success of that program, not only in southern Lake Huron, but even other areas of Lake Huron. Another question is, any chance of lowering the lake trout stocking to make room for more coho, steelhead, and Atlantics? What's interesting about this question, and thanks, Bryce, for bringing that up, is that one of our goals, overall, is to have a balanced predator-prey fish community. So we don't want to drive the prey fish to low levels and have the predators crash. And we recognize that we're dealing with a very diverse and ever-changing prey fish community. So our approach, and I would say a follow-up goal, is to manage a diversity of predators, from lake trout to coho to, as part of the reason for reintroducing coho, Chinook, Atlantic salmon, steelhead, et cetera. So, we were looking to changing, and by the way, we did reduce stocking of lake trout a couple of years back by approximately 66% lake-wide. And that was to account for some changes in natural reproduction. But I think some anglers in certain parts of the lake feel like that stocking reduction of lake trout was too far. And there's actually calls for reintroducing or restocking lake trout in areas where the fishery has declined pretty dramatically because the natural recruitment isn't holding up, and we're not seeing, obviously, the returns from stocking. So the overall answer to your question is, we'll constantly look at this diversity of predator, diversity of prey, and shoot for a balance. But we want to get away from managing a single species system like Chinook alewife dominated or lake trout bloater dominated, because we know the fish community is much more complicated than that, and a suite of predators and a diversity of prey promotes stability and health in the fishery. One of the additional questions was that was asked about thymidase and prey that is expressed in natural reproduction of predators. Is this factor, are there factors related to the quality of prey fish being considered the planting of predators? Yeah, this gets to, builds upon what I was just saying about a diversity of predators and prey. And alewife were a big driver in the thymidase deficiency syndrome that we saw across the Great Lakes, and played out in Lake Huron for sure. So, again, I think the solution, we're not going to be able to get away from this thymidase as it's now in the bacteria, in the food web through and through, but by managing a diversity of predators and prey, we're hopefully going to build a community that can compensate and mitigate for things like thymidine deficiency syndrome. And, we're seeing increased evidence of natural reproduction of lake trout and several other salmonine species across the lake. So good news, good question. And I know I kind of ran through those, and I could circle back, but other questions, Brandon, or ones that I might have missed as I was trying to follow through the question and answer session there?
No, that's great. Keep it rolling. If there's other questions, not just for the DNR crew, but other presenters that you didn't have a chance to ask, feel free to toss them in there. Randy, maybe as a teaser, you and it looks like Tim addressed the Cisco question. So early on, Greg asked, what attracts Cisco to Georgian Bay versus Lake Huron? So there's been this concerted effort to restore Cisco in Lake Huron, and we'll have Kevin McDonald from Fish and Wildlife Service share an update on that restoration stocking effort. But maybe you could kind of address that question about Georgian Bay in northern Lake Huron in the context of a teaser for next week.
Yeah, I thought Tim did a good job of kind of setting the stage of framing that in terms of, the habitat, the, what we understand about Cisco in northern Lake Huron, Georgian Bay, the idea that these stocks have been relatively stable, but they haven't really expanded to other areas of the lake. Cisco are unique because they can provide or serve as a prey fish at the younger life stages, but then grow outside of the sizes that can be consumed by fish predators. But then that leaves you kind of a bank of prey availability or production, because you've got a constant supply of adults, as well as providing fishing opportunities across a range of users. So yeah, getting into efforts to try to promote their colonization or building stocks in other areas of the lake, like outer Saginaw Bay, that is what will be discussed a little bit next week. And a lot of great work and a lot of great effort from our partners to help facilitate those stockings. And this is the early part of the program, so we're kind of looking for every little indicator we get on the returns of those stockings and so I encourage everyone to listen next week and hear about the updates. I know there was a question about creel clerks. Our creel clerks are working a full strategy this year. We have some temporary setbacks in our staffing because a lot of our creel clerks actually promoted to some other positions this year and are retired. So if you see gaps, it's because we're just in transition of staff, but not because of the pandemic. So we're moving on with trying to get a full complement of our creel program and estimating catch and effort. Looks like there's a question on could I briefly update us on the lamprey numbers and control efforts? By Mr. Ed. Yeah, so in sea lamprey control remains a priority for all the Great Lakes agencies. If you weren't aware, we had a crisis a couple years ago on the Ontario side. There was some pushback between some of the First Nations, the Mississauga and the Garden River First Nations, on sea lamprey control that led to some very critical streams not being treated for a couple years. So we're seeing some signals in the data in certain areas and certain species of minor upticks in sea lamprey, but we were able to reach agency resolve on that issue, and we will be aggressively pursuing an expanded sea lamprey control strategy for Lake Huron in this upcoming year, and anticipate that those numbers will be in check. But by chance you see something unusual or a large number of fish with transforming sea lamprey on predators that you wouldn't know, we love to get that feedback, and we'll pass it on to our counterparts of the Fish and Wildlife Service and Great Lakes Fisheries Commission to address those issues. In the data presented, why was Tawas not included? Many of the species discussed here are target anglers from that port. Yeah, so the port specific information, and by the way, Tawas is sampled both in fishery surveys. So crews have both bottom trawl and other survey information collected out of Tawas. And likewise, we do get angler input and feedback from anglers at that port. However, the creel program uses a stratified random design that basically doesn't sample every port and has some fixed sites and some reports that vary across years. So that's partly part of the reason why Tawas might not be included in our estimates of the creel survey, but we do have other information that you can reach out to us if you're interested. And we certainly try to expand or collect other information if we don't have creel clerks to every port. That's just a reality of the extent of the number of anglers and the amount of work and the resources we have to do creel surveys. So good to see those questions keep coming in, and certainly look for any other feedback, or if any of the other presenters, if you had questions about their presentations and want to raise those at this time, we appreciate all the feedback. And I would say we didn't really talk about it, Brandon, but again, I'll put another plug. I just saw a Michigan Tourism magazine come out and say that Lake Huron is the up and coming fishery. If you're going to fish anywhere in the Great Lakes, fish Lake Huron next year. And it was funny, they had a complicated survey. They also said, unless you want to ride roller coasters, and they suggested going to another lake. So I don't know how that fit in, but I do want to say, I think we're looking for some great opportunities coming this year. The uptick in the steelhead fishery was promising last year. I know this spring has been absolutely hard to understand, and it's thrown a lot of the anglers off, but I also have been hearing good reports at sites that normally wouldn't have runs, but we're hopefully going to see some increased fishing opportunities across the lake as we move into 2021.
Awesome. Thank you, Randy. Yeah, Lake Huron has, despite all of these challenges, remained a really invaluable fishery. And really, I think Frank Christ would say that we're really lucky to have a great DNR management team led by you, Randy, as the basin coordinator for Lake Huron, and just really appreciate all of the DNR's contributions to this session, as well as all of our presenters. We've, on Lake Huron, been really lucky to have a great core of research and management partners that are willing to have this conversation each spring with any and all who are interested in talking Lake Huron fish. So thank you to our presenters. And I need to thank, as we kind of round out our evening here, I need to thank all of you as attendees. Obviously, we'd love to do these workshops in person out in our coastal communities where we can have a more person-to-person conversation. But in the world we're in, this is the best we can do, and I'm glad we all were willing to get together to have this conversation, even if virtually. And for those of you attending as participants, we don't have this workshop if you don't show up and be a part of the conversation. So appreciate you all being a part of the conversation. I will make a couple of quick comments in closing. We will put an evaluation link. In fact, I think it'll pop up when you log off. So we'd love your feedback. We're always looking to find ways to make these workshops better, including looking for topics that you might want to hear about in future workshops. So if you have ideas, thinking like about the lamprey update question, right? Fish and Wildlife Service would surely be happy to share an update on sea lamprey management in Lake Huron in future years. So share your feedback. We'd appreciate that. And then a reminder and a promotional pitch for the next couple of weeks. 2 more Thursdays of Lake Huron Fish. Next week, we'll turn our focus to the nearshore fisheries. We're gonna, and this is Randy's great idea. We're gonna talk all these great fisheries of the St. Mary's River and the Lachino Islands area and this awesome Saginaw Bay perch and walleye fishery, all in one session, and cover some relevant topics like Cisco restoration and cormorant management that tie to some of those more nearshore fisheries. So,
Brandon, if I could just jump in, not to interrupt, but just another teaser for next week. So we will be discussing, or at least updating giving an update on the new permit system for managing the conflict with cormorants and fisheries. And also we're going to have Jared joining us, the new wildlife chief for MDNR. So, we're looking for a good discussion on that, and hopefully going to share some in-depth information about the new permit system, what Michigan is doing, and how we think that's going to provide a long-term balanced solution to this conflict going forward for Michigan.
Yeah. Thanks for adding that, Randy. And then the following Thursday after that, May 6th, the DNR Conversation and Coffee will be happening, and Michigan Sea Grant is super excited to collaborate with you all in making that available to anybody that wants to join. So with that, a lot of appreciation for everybody attending and contributing, and we'll do it again next Thursday. So I will normally say safe travels, but we'll just say have a good evening, and we'll hope to catch you in the next session or two. Thank you all.